polymerase chain reaction. Polymerase chain reaction is used to copy sequence of DNA in vitro, mimicking the DNA replication. As in replication, DNA polymerase enzyme is need to catalyze the reaction of elongation of the new DNA strand. Raw material needed are deoxyribonucleotide triphosphate (DNTPS), which are the 4 deoxyribonucleotides DATP, DGTP, DTTP, DCTP require single-stranded template DNA. Since the DNA polymerase cannot initiate DNA replication, primer is also needed. In PCR, primer is a specific sequence of DNA with small number of nucleotides, oligonucleotide, which is complementary to a sequence at 3 ends of target DNA to be copied. To copy both the strands to primers, each binding to either of 2 strands at 3 ends are used. In a cell, primer is an RNA sequence. In addition to this, Mg2+ is also required. These become the ingredients in PCR mix. DNA fingerprint, in which the sequence to be copied is present in dsDNA, and hence it needs to be denatured. Denaturation is done by heating the PCR mix to 95 degrees Celsius. At this temperature, most of the enzyme will denature. Therefore, it may be necessary to add DNA polymerase after denaturation. However, enzymes of the thermophilic organisms are resistant to high temperature. Therefore, the common heat-resistant DNA polymerase used in PCR is taqDNA polymerase, which is obtained from thermophilic bacterium Thermus aquaticus. Primer will bind to the complementary sequence of the denatured template DNA. This happens at a low temperature. This step is called annealing. Annealing temperature will depend on the sequence length of the primer. Once the annealing of the primer is achieved, then the prime extension DNA analysis will take place at different temperatures. This is the optimum temperature of the DNA polymerase used. When sufficient time is given, complementary copy of the template DNA is completed. At the end of this thermal cycle, denaturation, annealing, extension temperatures, one copy of each strand is produced. However, these are longer than the intended copy of the target DNA sequence. After a couple of PCR cycles, an exact copy of the target DNA is synthesized. After this, copies of the target DNA will be produced in an exponential manner that the 2, 4, 6, 8, 16, etc. After each cycle, after typical PCR will have 35 to 40 cycles. At the end, millions of copies of the desired DNA sequence will be produced from a single template DNA molecule. The repeating cycles are automated, done in a PCR machine, thermal cycle. PCR mixture is prepared in PCR tubes. These are inserted into the wells in PCR machine. PCR is the rapid method of obtaining a large number of copies of DNA with high accuracy. PCR is an essential technique in cloning procedure, which allows generation of large amounts of pure DNA from tiny amount of template strand for the study of a particular gene.